We often describe cancer as a tragic lottery, a consequence of getting older or having bad luck. But at the microscopic level, cancer is the result of a specific mechanical failure. It occurs when a critical internal security checkpoint is compromised. Among the various safeguards in our biology, the TP53 gene serves as a master regulator. It produces a protein called P53, often described by biologists as the guardian of the genome. Its role is to continuously scan DNA for damage and prevent defective cells from multiplying. To see how vital this single gene is, consider Li-Fraumeni syndrome. Individuals born with a defective copy of the TP53 gene face a 70 to 90% lifetime risk of developing cancer. Without a fully functional P53 protein, one of the body's most important barriers against tumors effectively disappears. While it is part of a wider immune network, functional P53 is a foundational defense, standing between normal cellular life and aggressive mutation. This guardian is not merely prone to accidental failure, it can be actively targeted and dismantled. Certain external viruses have evolved the specific ability to seek out and destroy our primary genetic defenses. High-risk strains of the human papillomavirus, or HPV, deploy a protein called E6 for this exact purpose. E6 acts like a precision tool, binding to the P53 protein and flagging it for immediate degradation within the cell. Once the guardian is removed, the infected cells can bypass their growth limits and form tumors. Recent laboratory research has identified a similar interaction with SARS-CoV-2. In these models, the virus's spike protein appears to suppress the transcriptional activity of P53, hindering its ability to activate the genes that normally force a damaged cell into lockdown. This has led to a scientific hypothesis about compounding threats. Researchers are investigating whether a chronic HPV infection, combined with spike protein exposure, could leave a cell's tumor suppression mechanisms doubly compromised. These interactions highlight a significant vulnerability in human biology. Our defensive architecture relies on specific genetic checkpoints that viruses have learned to exploit. Statistically, an animal with 100 times more cells should be overwhelmed by cancer. More cells should mean more opportunities for things to go wrong. Yet the African elephant rarely develops the disease, a phenomenon known as Petos paradox. Human cancer mortality typically falls between 11 and 25%. For elephants, it's just 4%. Genomic analysis reveals why. Humans rely on a single copy of the TP53 gene, while elephants have evolved 20 copies. Elephants effectively mitigated the risk of cancer by building massive redundancy into their security system, ensuring that if one copy of the guardian fails, 19 others remain on duty. When human DNA is damaged, our solitary P53 gene often tries to repair the cell. It is a slow, difficult process. If the cell struggles to patch the DNA correctly, it can accidentally allow a mutation to survive and divide. Elephant cells use a more aggressive strategy. Instead of risky repair, their multiple P53 copies instantly trigger apoptosis, or programmed cell suicide. This is a policy of zero tolerance. By instantly vaporizing the microscopic threat, the elephant prevents a single damaged cell from becoming a tumor. In nature's most resilient species, the ultimate defense isn't cellular healing, it is the immediate destruction of the threat. While we cannot naturally duplicate our genes, modern oncology is beginning to mimic the elephant's defensive strategy. New drugs currently in clinical trials, called MDM2 inhibitors, are designed to stabilize our single copy of the P53 gene. The goal is to awaken our natural guardian, allowing it to more effectively recognize and eliminate existing cancer cells. In the meantime, the most practical defense is protecting the genetic guardian you already have from the viral degradation we've identified. This involves specific, actionable steps: staying current with HPV vaccinations, attending regular screenings for early detection, and taking measures to minimize exposure to chronic viral stressors. Long-term health is a combination of our natural genetic capacity and proactive preventative medicine. It is important to contextualize the viral hypotheses mentioned earlier regarding SARS-CoV-2. These interactions are currently based on laboratory models. There is no clinical evidence at this time suggesting that COVID-19 or its vaccines increase cancer risk in the general population. Your biology contains a sophisticated system of checks and balances. By understanding and protecting these foundational genetic mechanisms, you are simply giving your body's natural defenses the best possible chance to succeed.